WHONET and One Health applications. So the agenda will cover 4 main areas. Reducing my controls so I can see my slides better. Great. 4 main areas. Antimicrobial resistance from a One Health perspective, some high-level concepts. Then the rest of it will be dedicated to software and data management. I will not do a deep dive into the very complex and rich issues of antimicrobial use and resistance outside of the human health sector and integrated surveillance. And I apologize in advance if I make mistakes. Feel free to make corrections and additional comments in the chat. The focus of this will be the software and data management after we introduce some of the high-level concepts. So the second point is our breakpoints. Clinical breakpoints, microbiological breakpoints, human breakpoints, animal health breakpoints. Followed by some chat, but then software demonstration of WHONET and some of its features related to One Health, animal-specific, food-specific, integrated across sectors. And then finally, backlink, importing data from a variety of systems to get the data into WHONET for purposes of analysis. One thing I want to say is that the things that we cover for animal health are often extremely relevant on the human health side as well, but they're often less needed. One of my main points is that in the human health side, most hospitals in the world basically have the same kind of information: patient name, maybe date of birth, age, location, specimen date, specimen type, organism antibiotic results. In WHONET, we give people the standard defaults, and most people are pretty happy with that. They can change them if they would like. On the animal health side and the food and the environmental side, there are so many objectives, so many needs. So they have the same features that the human health people have, but they actually utilize them. On the human health side, people often do not change very much the defaults. They do if there's a research project or some special need. But outside of the human health sector, the needs are much more diverse and the data management collection needs. So some initial high-level broad concepts. Here we see the world. We see people, we see animals and fish and water and land and farmland, and all of this is related. The microbes move between them. Here you see the red dots in the image. The red dots represent antibiotic residues. The antibiotic residues are given to the antibiotics are given to the humans and the animals. And then from there, they do spread around into the food, sewage, water, and impacting the environment, and eventually back to people, and eventually also to animals. One thing which is not explicitly mentioned in this slide is also industrial uses. Antibiotics can also be used in industry manufacturing, which is mentioned on my next slide. So antimicrobial drivers and selection and spread, it's complex, and these are all One Health. And a change one place, an inappropriate use of antibiotics in one sector can impact everything. The microbes appear in one sector, in one part of the world, in one usage scenario, and from there can spread into the One Health concept. So there are diverse data needed at all levels: data on resistance, data on use, risk factors, food production practices, healthcare infection control practices, regulatory interventions. This is a nice visual image. This one is what Rebecca Irwin from Canada has referred to as the confusogram. There's so much going on here, but it is a reflection of what is happening. Many of us focus in the lower right-hand corner of the screen in that human area. Hospitalized humans, community, long-term care, short-term care, travelers, and their role in moving things between continents and between countries. You see food animals, lower left, aquatic, upper right. Top left, you do see examples of industrial use in ethanol production, distillers. I've seen it used, for example, in, I think, underwater pipes, just to decrease, if possible, cloggage within the pipes. And again, if I do make mistakes, apology in advance. I am not an expert. This is one of my normal slides that I would generally use, but I do want to highlight some of the differences between human and non-human sectors. You have 4 broad categories of objectives: policy and advocacy. epidemiology of resistant microbes, basically that core underlying scientific understanding. Resistance containment, including treatment guidelines, outbreak recognition and response, and capacity building, laboratory capacity to ensure laboratories are doing the test correctly and reporting correctly, and epidemiological data management capacity. On the human side, we spend a lot of time in that resistance containment area. Not exclusively, but most of what we do on the human side are treatment guidelines, isolating patients with MRSA, with CRE, VRE, early detection and response to hospital outbreaks, and community outbreaks. So most of our effort on the human side is clearly in that resistance containment area. This also happens, of course, in the animal health area. Food animals, cattle, sheep, chickens, fish, and home domestic animals, companion animals, and wild animals and zoo animals. All of these have responsibilities. There are responsibilities in all of these for making sure the animals are healthy. In addition, there are these elements of food safety. We are eating many of these food-producing animals. There are regulatory aspects. Is the food system safe? What are contamination levels? Is there resistance coming from the food sector? If yes, what is the impact of resistance in the food sector on human health? Is it a tiny amount, 1%? Is it a massive amount, 80%? So on the animal health side, there are often issues of uncertainty. Does this happen? Does resistance cross over? Yes. But what is the impact? Is it a small impact or a large impact? And we need to develop risk mitigation, risk analysis, risk assessment strategies to say, well, these are risk factors, but what do we need to do about it? And this is where there can be controversy, differences of opinion, especially when you consider the priority needs of public health authorities, food producers, industry players in market distribution. So I'll just talk a little bit about that. A related aspect is on the human health side. 99.9% of the data from humans comes from sick individuals. They go to health clinics, they go to hospitals, they complain of their symptoms, urinary tract, cough, etc. A sample is taken. On the animal health side, there's a very large proportion of sick animals, but also healthy animals done to ensure food safety. Either in a regulatory manner that's ongoing, routine collection on a yearly basis or a monthly basis for purposes of monitoring, and also periodically research projects. Let's just do one study to see in the healthy animals what is going on. Of course, on the human health side, who pays for the tests? Well, typically the patient pays for the test, or the insurance company pays for the test, or the national government health system pays for the test. On the animal health side, if it's a diseased animal, often the farmer would pay for the test. If it's a healthy animal, who is going to pay for that? And that's a problem because many times no one pays for it and it doesn't happen. On the human side, of course, we have issues of bias. Many patients treat themselves, they get better. They come to the clinic and we treat them, but we don't take a sample. We take samples from the most complicated patients. We take samples from the ICU patients. So there are significant biases, very important biases in the human health sector. They're often even worse in the animal health sector because often there are practically no or very little routine clinical samples from sick animals, unless if it's many of the animals will kill or we'll just treat them with 3 or 4 or 5 antibiotics. If it's a companion animal like a cat or a dog, or a large animal like a cattle, a cow or a horse, it's more likely we'll take samples then. But they're often very biased of animals who have not responded to therapy, and often in very small number. And to make treatment guidelines for diseased animals, you often want to collect data from healthy animals to get an unbiased representation. But who is going to pay for that? We do some sampling of human patients as well, but it's just to a much lower degree than in the animal sector. The animal sector, this is often referred to as, it's a little bit inaccurate, but to a large, it's mostly accurate. We're talking about passive surveillance. These are the routine samples that come in, and the routine samples in many settings are simply the diseased animals. These samples will come in simply as part of routine animal health care. And then we talk about active surveillance, where people are paying for the collection of data from the healthy animals. That doesn't have to be the case. You can do active surveillance on diseased animals as well if the sample was not going to come in. So you do need to be a little bit careful of the meanings, and sometimes the meanings have different, it can mean different things to different people or in different contexts. So I'll just give one example here, regulatory approaches for restricting the use of growth promoters in food-producing animals. We give antibiotics to people primarily for one reason. No, I'd say two reasons. The person is sick. This is therapeutic use. Or prophylaxis. They've been in contact. They've been exposed to somebody, for example, with bacterial meningitis. We might do prophylaxis. This is also true on the animal side. We have therapeutic, treating diseased animals. Prophylactic for treating individual animals at high risk of getting sick. There's a kind of prophylaxis known as metaphylaxis where you do this more at a herd or a group level. Again, disease prevention is the objective. And then you have the issue of growth promotion. The animal's not sick. Why do we give antibiotics to animals to make them bigger? How do we get a bigger chicken? One, take better care of the chicken. Vaccines, more nutrition, more food. Not too expensive. What we can do is we can give a little bit of antibiotic. Some antibiotics will just kind of decrease the microflora and microinfections, so more of the food the animal consumes becomes meat. Or I think some antibiotics, I'm not an expert, do have a little bit of a hormonal impact. But the goal of growth promotion is not disease prevention. It's simply more meat, better food production, economics, and it's a controversial practice. So in Europe in 2006, after many years, starting in Denmark and other countries, with voluntary bans on growth promotion of antibiotics also used in human medicine, and then all growth promotion, antibiotics as growth promoters were banned on the basis of their precautionary principle. Using antibiotics for non-therapy health reasons doesn't seem necessarily like a good idea. We have evidence to suggest it might not be a good idea. We have evidence to suggest that lack of growth promotion does not harmfully impact the economics or the animal welfare. So on that basis, Europe decided to ban growth promotion in the year 2006. The United States, we faced a number of similar issues, but we approached this from the principle of demonstrated harm. So we do not ban growth promotion here in the United States at this we have not done that. Approximately 1995, enrofloxacin was a fluoroquinolone, was approved for use in chickens in the United States as a growth promotion. It was permitted with the understanding, with the requirement that the U.S. establish a national program known as initially as NARMS Enteric, now simply known as NARMS, National Antimicrobial Resistance Monitoring System, to look at surveillance between the human, animal, and food sectors. The U.S. CDC, U.S. FDA, the USDA, the Department of Agriculture. And after 5 years of data monitoring, we looked at the data. I was very involved in the USDA part for 5 years. And after the first 5 years, we looked at all the data from the Salmonella, and we found fluoroquinolone resistance did not increase. Great. Campylobacter resistance rose very quickly. So on that basis, the FDA did not ban growth promotion in a general sense. They did ban the use of fluoroquinolones in chicken and poultry feed in 2005. So we had the same underlying discussions as happened in Europe, but we came to a different conclusion in terms of risk assessment and risk management and risk mitigation. We've had a proposal for a law called STAR, Strategies to Address Antimicrobial Resistance. I looked last night, I found there's another one which has also been proposed called PASTEUR, Pioneering Antimicrobial Subscriptions to End Upsurging Resistance. But these have been before Congress but have not passed. Another high-level issue, confidentiality. Confidentiality, of course, is important. But the issues are different. When we talk about human health data, we especially think about protecting the person's name, their date of birth, their address, phone number, medical conditions. The patient is utmost in our mind. In the US, we have a law called HIPAA, protecting 18 identifiers or 17 identifiers that are considered to be confidential. There's also healthcare institution confidentiality. Which hospital, you don't want to be known as the hospital with bad MRSA rates or bad infection rates and bad outbreaks. And you also have quality. You don't want to be known as the hospital with the bad quality laboratory. Then there's societal confidentiality about cholera, and there was one example of New Delhi metallo-beta-lactamase, NDM1. And the fact that New Delhi was in the name also did create some issues about blaming certain areas for resistance. We have very important issues of confidentiality on the animal health side, but the confidentialities are different. The animal doesn't care. We're not trying to protect the feelings of the animal. But especially in a food production setting, the confidentiality of the farm, the factories, the slaughterhouses or the abattoirs, the distributors, and the country. There have been some nice studies from EFSA, the European Food Safety Agency, where Northern Europe had low resistance and low use. And I'm talking in this particular example about swine production. Southern Europe had high use and high resistance. And then there were countries in the middle that had low use and high resistance. Why was that? Because many of the animals were imported from Southern Europe. Is there a health issue there? Well, that's an important question. But it also became a political question with protesting farmers. We need to protect us. Part of it is a protection of the health of the country, but part of it is also protecting the economy and using this as a mechanism to say that we should not be importing food. So there are very important issues about confidentiality related to economics and marketing and protecting patients, and then the industry element. There's also non-food producing animals about rabies and what communities, what cities have rabies issues. Doesn't mean to hide these issues, but it does mean appropriate people need information to take appropriate action. Environmental, you don't want to be known as the river or the lake or the water, the soil area. So you do need to consider what is required by law to report. What is a good idea to report? Who should know about it? At what level should be known about it? In a different context, FAO InFARM, that will be the subject of our next webinar in June, offers countries 3 levels of confidentiality. One is you can give your data to FAO, and they use the data internally in an aggregate way and for certain things, but you're not publicly, the country is not named. And other ones, you can say, well, yes, please use my data, but don't, and you can distribute my data, but don't tell people which country is which. The third one is, yes, you can be on the map, your country is labeled, your aggregate statistics are labeled. So they've given the country the option to decide what their comfort is and their regulatory requirements are for reporting. That's a little bit of the end of my high-level introduction to resistance, a very complex topic. Let me just take a quick look at the time. Great. I was very involved for the last several years with the Fleming Fund, especially the WHONET Strategic Alignment Grant, Captura for Asia, Radar for Asia and Africa, and also providing a supportive role to a number of the other Fleming Fund grants, including AMRO. There were 4 regional grants for antimicrobial resistance in a One Health perspective, One Health context. So one of the things they did in that is a presentation on integrated surveillance systems. And I found this valuable article published in 2024, the reference is down below, where they tried to see what is available. What are some models and what are the challenges and success stories for national integrated surveillance programs? They looked at integration in 2 levels. Information integration, what data sets were brought together? Human, animal, food, was that integrated? Fish, cattle, chicken, antimicrobial use. genomic aspect, clinical diagnosis aspect. So they looked at the integration of the information in databases. They also looked at structural integration. Did each sector work separately? Did they communicate on their data, on their reports, on their announcements? So they looked at integration of supervision, data collection and management, data analysis and interpretation, and then communication and dissemination. They collected all these data, they did a cluster analysis, a hierarchical cluster analysis, and they identified statistically 4 groups. But before I mention the 4 groups, the first thing you will notice on this slide is every one of these is a high resource country. So the first conclusion from this paper is there is obviously insufficient information about what is happening in low and middle income countries. Partially that reflects that there may be no activities. Often it reflects that there are activities that have not been published in the international scientific literature, which is very unfortunate. When I saw this article, I said, but they left out Vietnam, they left out Bangladesh, they left out countries with very important activities because I don't know whether or not there were journals, but they did not meet the criteria for inclusion in the systematic review. So the first conclusion is we have some nice examples here from high resource countries. Are they relevant for low resource countries? Getting back to my earlier comments about growth promotion in Europe, they decided that in their context, growth promotion was not needed for a healthy, productive, economically valuable food production sector. I just don't want to make that same conclusion on a low resource country because it's a different set of dynamics. It'd be excellent to do these kinds of research to demonstrate what is the value of growth promotion and these other mechanisms about vaccination and food and cleanliness and hygiene. So much of the data needed for this comes from a high resource country, and we cannot simply say that the same conclusions would apply in other settings. So we found 4 groups. The group that I put at the top is the one that's the highest degree of integration. There's one database. I have a visitor leaving. Thank you so much, and we'll catch up next time. And so we have the highest level of integration, a single database. You can go to that database and you have the human, animal, food, maybe antimicrobial use as well, all in the one database. United States is there, NARMS, DANMAP, NORM, Sweden. Group 2 is each group works separately. You have the human sector, the animal sector, the food sector, maybe the environmental sector. They work independently, but they collaborate at the higher levels. They may share reports, they may share tables, they may share communication strategies. And you see, for example, the Netherlands, the UK system, Japan, France, the Observatoire National, the National Observatory. Canada has all sectors, human, animal, food. I don't know if they've started environmental. It's all 3 sectors, but the coordinators are on the human side instead of an equal partnership as it is USDA, CDC, and FDA that I mentioned. And group 4 was led by the food side, generally not including human isolates, but food and animal isolates. The ones in green have an interactive data portal. I've included these interactive data portals on the next screen. I won't go into that into any detail, but you might find them valuable later. These that you see here are from the systems described in that article I mentioned. Not mentioned in the article, WHO GLASS on the human side, InFARM, these are all United Nations on the food and animal side. GISA, I don't know its current state of development. This initially was known as TISA, the tripartite, integrated surveillance system of antimicrobial resistance, I guess. I don't know where the R went. But GISA is, just generalizing it. It was 3 organizations, WHO, FAO, and what used to be known as OIE, now known as WOA, the World Organization for Animal Health. It was 3 organizations. Now it's 4. So it's a quadripartite, including the UN Environmental Program. G simply means global. That way, if they decide to add 5 or 6, at least G covers it all. The Fleming Fund AMRO projects. I recently attended the closing session of the AMRO South Asia group, and there were excellent presentations there, excellent reports that came out at the end, and it just demonstrated that a lot of countries had done a lot on the human side. a lot on the food and or animal side. And in many cases, they had never really integrated before to see what are the resistance findings, what are the organizational challenges, where do they get their discs? So it was a valuable capacity building exercise, but for many countries, it was the first time they really could bring the data together to highlight areas for future coordinated messaging. I mentioned Vietnam, Bangladesh. Resistance Bank is from a group in the United States, One Health Initiative. The old name was CDEP, I believe it's One Health Initiative. And it's basically a literature review of existing data, data from publications. So that's the end of the first portion of my presentation. Adam, any questions right now in the chat?
No, I don't think so, John.
Okay, thank you. Now everything else is going to be data related. Okay. So a common question is what breakpoint? I get the question all the time. John, I'm working in an animal lab. Which breakpoint should I use? Easy question, difficult answer, complicated answer. Should I use the clinical breakpoints or these things called epidemiological cutoff values? So let me talk about clinical breakpoints. There's a common misperception, perhaps by many people on this call, that the reason we do susceptibility testing is to find resistant bacteria. That's sort of correct, but it's not exactly correct. What do I mean by resistant bacteria? If I mean bacteria with resistance genes, that is not the purpose of the disk diffusion or the MIC test. The purpose of disk diffusion MIC testing primarily is not to find bacteria that have resistance genes. So what is the goal? The primary goal is to guide clinicians into finding effective therapy, treatment options. We have sick people, sick animals that need to be treated. Which antibiotics would work? And why is there a difference? And that would be, I think, here. So a laboratory finding of susceptible by the clinical breakpoint for an antibiotic suggests that most patients, most human patients, animal patients, should respond to therapy, even if the bacteria has a resistance gene. Many resistance genes are weak. They're not expressed strongly, might be a single copy. And in sufficient doses, we give high doses, especially for things like urine, where things get concentrated. A bacteria that's a little bit resistant can often be effectively treated. So we might call the bacteria microbiologically resistant, but clinically susceptible. So the goal of CLSI and UCAST and other guidelines is not to say yes or no, this bacteria has a resistance gene, but it's to tell the doctor this drug probably will work or this drug probably will not work. And by work, I presume there are other things. For example, if the patient shows up at 1 o'clock in the afternoon and the patient dies at 1:15, the patient did not die because of antibiotic resistance in that case, because they just got there. So it has to do also with the severity of the condition when you started the antibiotics. But in normal circumstances, if you give an antibiotic to which the bacteria is susceptible, under most circumstances, the animal, the person should get better, even if there's a little bit of resistance. A good example of this is a lot of enteric pathogens. Salmonella, the main treatment for salmonellosis is oral rehydration fluid, bed rest fluid, fluid replacement, electrolytes. In these simple cases, I'm not talking about complex cases like Campylobacter and invasive, dysentery, Shigella, but in simple cases of watery diarrhea, it doesn't really matter if the bacteria is resistant or not. The patient's going to get better eventually. You just need to do oral rehydration. So that's an extreme example where the susceptibility is not the most important thing. Similarly, if somebody has a skin infection with a Staph aureus, the main thing the person often needs is soap and water, good hygiene, protect it, and the person will eventually get better. The antibiotic will help people to get better more quickly in many cases. So this is the idea of clinical susceptibility. The animal, the person should get better, even if there's a low degree of resistance. In contrast, finding a resistant means that the animal, the patient, probably will not get better. There's a high risk of treatment failure. There is what we call the in vivo in vitro correlation. Let me switch that. In vitro, meaning in the laboratory, in vivo correlation. A high degree of correlation means if the lab says sensitive, the antibiotic should work. If the lab says resistant, the antibiotic should not work. There is very good in vivo, in vitro correlation for things like malaria, tuberculosis, shigellosis, um, uh, other things like respiratory infections. If someone has an ear infection, even if the bacteria is resistant, they often still respond because many patients will respond anyway eventually, just the natural immune system and drainage of the internal drainage and the urine when antibiotics get concentrated. So in vitro, in vivo correlation is important for assessing the clinical impact of resistance. So the likelihood of successful clinical outcome in part depends on the bacteriology. That depends on the microbe's characteristics, this idea of microbiological resistance. But it also depends on the pharmacokinetic, pharmacodynamic parameters of the infection. And these may depend on a number of factors. For example, if a patient has meningitis and the bacteria is a little bit resistant, the patient may die. The antibodies often do not cross the blood-brain barrier very quickly, so a little bit of resistance might be extremely important for meningitis with death. But if the patient has an ear infection or even pneumonia, septicemia, it's in the bloodstream, so the concentrations that are given immediately very quickly reach the sites of infection. So the clinical susceptibility resistance depends not only on the characteristics of the organism, it also depends on the infection. Is it in the urine? Is it in meningitis? Is it in the ear? And that also, if you extend it, can extend to the animal. If something is a cat or a dog or a fish, a little bit of resistance may or may not be important. In fact, CLSI has a few examples where they do provide different breakpoints for young horses or adult horses. I'm not certain personally why they came to that decision. It might have to do with kidney function and concentrations. It might have to do with natural, intrinsic immunological competence. Maybe an adult horse has more immunity or is less susceptible to dying suddenly. I don't know the reason, but for CLSI, there are a few young horse versus adult horse. It's not microbiological difference. It's a PK/PD host difference. And that's why breakpoints in CLSI and UCAST have become more complicated over time, recognizing that our goal is not finding resistance genes, is to give the clinician some guidance as to what antibiotics should work. The clinicians are not doing research, they're not doing pharmaceutical clinical trials, they're not doing molecular biology, they simply have a sick person, a sick animal, and they just want to know, is this antibiotic probably going to work, even if there's a little bit of resistance? There's this other concept about epidemiological cutoff values. One of the very first presentations I ever went to was in Norway at an ECCMID meeting, and J.D. Williams was talking about the concept of microbiological breakpoints. I like the word microbiological breakpoints. I think it's clearer, but it's the same concept as epidemiological cutoff values. And his proposal was that there should be a breakpoint that can distinguish phenotypically. I'm not talking genetically. Genetically, you just do the genetics, the genomes, etc. But from a phenotypic perspective, can you distinguish reliably between bacteria that do have a resistance gene to ciprofloxacin versus those that don't? If they don't, we call that non-wild type. If they do, we call that, if they don't have a resistance mechanism, we call it wild type. If they do have a resistance mechanism, we call it wild type. If they do have a resistance mechanism, we call it non-wild type. So UCAST developed this concept of microbiological breakpoints further and have called it epidemiological cutoff values. The Europeans have called it ECOF, and the CLSI has called it ECV. They mean the same thing. I will call them ECOF simply that's because they've done a lot more work in publication of their ECOFs. It's also 2 syllables instead of 3 syllables. So in this presentation, I will call them ECOFs, but they mean the same as ECV by CLSI definitions. So the goal of ECOFs is not predicting clinical outcomes, but rather discriminating bacteria that have or do not have phenotypic evidence of a resistance gene. An ECOF is a characteristic of the microbial species and is not impacted by animal species or clinical syndrome or other PK/PD considerations. For bacteria with a low degree of resistance, and I gave some examples earlier, it would be common for an organism to be quote unquote microbiologically resistant, which was J.D. Williams' terminology, but still clinically susceptible, meaning the antibiotic should still work. So which breakpoints should I use for reporting and data analysis? Well, some people say, well, if I'm working in animal health, I should obviously use the animal breakpoints. They say, well, it's not so obvious, and I'll give examples. So when should we use human clinical breakpoints? Well, obviously, if you're working with human samples and diseased humans or getting data from healthy humans to make treatment guidelines for sick humans, using the human clinical breakpoints makes perfect sense, obviously. The large majority, over 99% of data around the world are presented with human breakpoints. In the human setting, but also in the animal setting, it's very common to use the human breakpoints, and I'll comment further why. Human breakpoints are also relevant, as I said here, in the animal food environmental samples, not because the animal is sick. Well, the food is dead. The environmental sample, there's no life in that. But the goal is not curing the animal or the food or the water. It's an assessment of what would be the direct clinical impact on the human population. So it's what we might refer to as a public health concern. So it is appropriate to use human clinical breakpoints in animal and food and water samples if our objective is to inform the public health assessment risk for human populations. It's also helped to facilitate, if you want to compare the human, animal, food data, it makes sense to use the same breakpoints across the board. Otherwise, you may come up with conclusions that have more to do with the breakpoints than actually the microbiological differences. So human breakpoints make very good sense if you're working with human data. They also make good sense if you're working with animal, food, environmental data, if one of your objectives is contributing to the discussion about the human health risks. Animal breakpoints. These are appropriate when you get samples from animals, whether they're healthy animals or diseased animals, or food samples or environmental samples, where the goal is the development of treatment guidelines or assessment of resistance, emerging resistance in diseased animals. So if your objective is to take care of sick animals directly for that animal or indirectly through the development of treatment guidelines, using the animal-specific breakpoint makes sense. And finally, and I misspelled it, epidemiological cutoff values, appropriate for use in any sector, human, animal, food, environmental, if your goal is the phenotypic recognition of which strains do or do not have resistance mechanisms. And this can also be done across sectors. So I mentioned if you want to go across sectors, many people choose to use the human breakpoints in order to have common benchmarking. The other approach is just to always use ECOFs. In that case, you're not worrying about the PK/PD and the treatment outcomes and the guidelines. You're simply looking at the microbiological differences. So if you want to go across sectors, then using the human clinical breakpoints or the ECOFs, either approach could make good sense. If you have a focus on clinical outcomes in human, I would go with that. If your focus is finding resistance genes, collecting samples to look for genes and do genome sequencing and molecular typing, then the ECOFs could have value there. For example, in EARSnet, the European Surveillance Project on Human Isolates, of course, they use the human clinical breakpoints. EARSvet, the much newer program on the veterinary side, they look at the ECOFs, so they can look at a microbiological comparison. Because their goal, their objective is not developing treatment guidelines immediately, they look at the ECOFs. Okay. Conclusion, all are relevant. So the answer is, if people ask me which breakpoint should I use, I often say you need all of them. Today, use the human breakpoints. Tomorrow, use the ECOFs. The next day, use the animal disease breakpoints, depending on what your objective is in that moment. And in WHONET, you don't need to decide at the time of data collection. It's an analysis issue, and WHONET dynamically will analyze with what you say. And in fact, it does all of them at the same time, and you can choose to filter on it if you only want one of the three. So all are needed, and we handle it dynamically, so you do not have to make the decision at the time of data collection. However, to use these, you do need to know the zone diameter or MIC measurements. How many millimeters? The ECOF for zone diameter, the clinical breakpoint might be 20 millimeters, whereas for susceptible, and the microbiological breakpoint might be 16 millimeters. So, for example, 1718 might be microbiologically resistant with a resistance gene, as suggested by the phenotype, but it would be clinically susceptible. I think I got my numbers backwards. That's right. That's right. 20. I got the numbers backwards. 25 might be microbiologically susceptible, but 20 might be clinically susceptible, accepting even a small degree of resistance. So apologies for my confusion there. If you just collect RIS category data, you cannot dynamically or statically reinterpret the data. So anyone on the human health side, we strongly recommend collecting the measurements. We recommend that very strongly in the human health side. It's even more important in the integrated health or the non-human health sector, because you do need to apply these different breakpoints, ideally. So status of breakpoint development. So you might decide, I want to use the animal-specific breakpoints, but you got a problem. CLSI is very well developed on the human health side, M100, M45, a number of these other documents for bacteria and for fungi. And they have also a lot of animal breakpoints, animal clinical breakpoints, but especially companion animals, cats and dogs, horses, and some bovine mastitis. They do have a couple of chicken, a couple of sheep, but even if you're in, so on the animal side in CLSI, there are a large number of breakpoints, but it's mostly for cats and dogs. So on the food production side, we really don't have a lot of breakpoints. So many times people use the human breakpoints because they're the only breakpoints that are available, unfortunately. And CLSI does have ECOFs, ECVs, but very few, very specific situations. So I can look at the time. Good. UCAST, very well developed on the human side, human clinical. The animal clinical breakpoints, they have a committee called VetCast, and there have been a very, very tiny number of publications of published breakpoints. If I have time, I might show the website, but in Europe, many clinical labs, veterinary labs use the CLSI breakpoints, clinical CLSI breakpoints, because they exist, and there just are not sufficient number. I don't know the current status because I've joined webinars and there are many proposed and approved, but just on the UCAST, the VetCast website, there are very few published there. And then, but UCAST has done an excellent job with ECOFs, both on the MIC and DISC side. They're undergoing reviews and revisions and evaluation. They changed their criteria for what acceptable data contributions are. So they publish ECOFs, as well as TECOFs. TECOFs are basically the old ECOF, but tentative, given that they want to revisit it with applied standards for quality and methodology and number of isolates, et cetera. Ongoing work. Those are available. They're all available on the UCAST website. One bacterial species at a time, or one antibiotic at a time. Unfortunately, you cannot download an Excel file. So we've been discussing with UCAST, can they try to get that up onto a website? Because these ECOFs are so important, but they're not that convenient in mass. You have to look them up individually. Because of the incomplete availability of breakpoint, many people will only use human breakpoints or ECOFs because they're just not sufficient animal-specific breakpoints outside of dogs and cats or bovine mastitis. For animal clinical breakpoints, CLSI is good, but as I mentioned, just a few priority species. For ECOFs, UCAST is an excellent option, and these should work for CLSI testing. The methodologies are similar enough that you can apply European breakpoints and European ECOFs with some caveats to the experts on the call. But generally, you can use the breakpoints as long as the disc diffusion potencies are the same. If CLSI uses a 5 microgram disc and UCAST uses a 5 microgram disc, you can use the values. If CLSI has a 5 microgram disc, but UCAST has 20 microgram disc, you cannot use those numbers. Make some comment. WHONET has our old way of doing things and then the new way. I created the old way myself 30 plus years ago. And at that time, for anybody that old, there were these things called the general breakpoints. At that time, it was known as NCCLS, the National Committee for Clinical Laboratory Standards. And in Europe, before UCAST, the breakpoints was one long piece of paper. All the breakpoints were on one page, and there were a couple of exceptions for strep pneumonia and Pseudomonas. Besides that, everything was a general breakpoint. And I had lunch last week with Tony Medeiros, and in the 1960s, he originated this concept of species-specific breakpoints. He said, the Proteus, it just doesn't match the E. coli and the Klebsiella. And CLSI UCAS came to the same conclusion. So around 2000, CLSI and UCAS, UCAS was a merger of several other European systems, when completely they dumped the general approach. Every bacterial grouping should have its own species. So the old approach, we had these tables, but also it was mixed in with stuff that I had hard-coded into WHONET. So if somebody asked me for the breakpoints, I could give them the breakpoint tables, but I just couldn't share with them the internal stuff I did about meningitis and cats and dogs. It was all hard-coded into WHONET, or special rules like MRSA, are resistant to most beta-lactams automatically. Linear beta-lactamase negative, ampicillin-resistant Haemophilus. These were all special rules that were hard-coded into WHONET and therefore could not be shared with others. The new approach, everything is in these tables, and Adam has done this work. We did this in collaboration with MSF to support their mini LIMS project, where we have these tables that you can download the tables if you have interest in this. And Adam, maybe you could put the link into the chat if anyone might be interested in that. I thought about it for the slide and I forgot about it. So you can and actually you do have them when you install WHONET, the tables are there. We also keep them on a separate website for people who don't want WHONET, but they just want the tables. So mini LIMS, the MSF LIS, to accompany their mini lab project, they use the WHONET software for analysis, but they also use the WHONET tables to do interpretations. Vietnam does as well, and a few other groups use the WHONET tables for their own purposes. In the past, they tried to do that, but they didn't have all the special rules that I mentioned. Now it's in these tables. They've been available in WHONET since approximately 2000, but we switched by default until 2025. It took us a while because we had to do some things with user-defined versions of this. But when you do new lab in WHONET, automatically you get the new approach. We do encourage you to switch from the old to the new for many reasons, but especially in the food and animal sectors. So how do you upgrade from the old to the new? WHONET will ask you, do you want to upgrade to the new approach? And just say yes. Or you can do it manually. You can decide, go to data analysis, go to lab configuration, antibiotics, breakpoints, and it gives you the option about data-driven tables. I'll show that during the demonstration. So WHONET, I've shown you most of the PowerPoint. I'm going to go through this relatively quickly because I would rather show it to you as a live demo. WHONET is widely used around the world, as all of you know. I do want to highlight at the bottom, we sent out last week and a reminder this week about this map that you see in front of you. This map is from 2013. I need to update this map. So we look for your assistance in just getting back to us about how many labs, which countries. Like even when I look at this, I know some countries that are not listed here should be there. So it's only got about contact information, what year did you start, how many labs, if you're a network coordinator. We only need one response per network. And then we also were asking about any suggestions about WHONET, new development, new areas of work. We were also asking for testimonials. We're trying to promote WHONET out there. So if you just have some nice stories that you liked WHONET and some value, you can give us a testimonial through the survey. Adam, we could also put something on the WHONET website where people can write a testimonial. We would not distribute that without your permission, but we would like to collect these testimonials because they would be of value to us. This part very quickly, WHONET is configuration, data entry, data analysis. We've covered that in previous WHONET webinars, covering everything, but often with a focus on the human side. But I'm going to basically do a lot of the same thing, but focusing some of the non-human elements. The last point on here is about InFARM. InFARM will be the subject of the next webinar in collaboration with FAO, and the tentative date would be at the end of June. Okay. Laboratory configuration, you choose your antibiotics, your locations, your data fields. I mentioned the veterinary breakpoints are there, the veterinary agents are there. Locations, I'll show that on the demo. Very important data fields. I started to mention that earlier. On the human side, the questions are pretty stereotypical. First name, last name, date of birth, optional, age, patient location, specimen date, specimen type, organism, antibiotic results. Some people add things like date of admission is nice if you have it, diagnosis, name of the doctor, gram stain if you're using WHONET for clinical reporting. So most human labs don't change the list, or they only change it a little bit. On the other hand, the animal labs often change this significantly. We give them a different list by default. We don't ask the animal's first and last name. We don't ask the date of birth. Animals have a date of birth, but people don't usually know the animal's date of birth. So WHONET, we have automatically removed fields that we don't think you need, but we've added other fields like animal species, production type, and you can add other fields. And many of you have very long lists, and others of you have very short lists. So on the human side, people often don't change the list very much, but on the animal side, they do. Or research side, research projects, they also change it a lot. Data entry, at the top, you choose human, animal, food, environment. I'll show this during the live demo. You can import data with backlink, and that's covered in another part of this PowerPoint. I'll skip over this slide. These are some examples of data analysis. I leave them in my PowerPoint for the benefit of people who might want to download my slides, but don't want to listen to the 2-hour webinar. So I'm not going to spend time on this right now. These are output to the screen, to Excel, to DHS2, to Word, PDF. Interoperability, we can import data from many systems with backlink. We can export data to many systems, including GLASS and InFARM and EARSnet and now EARSvet. Here are some specific adaptations we have done for One Health. We started really with the animal health in around 2000, and it wasn't 2001, I went back, it was 2000. We started with the NORMS project then. So our original work was US-based, USDA, FDA, CDC. And then later with WHO, later with FAO, and obviously FAO has been a primary contact for the last several years in this area. We're starting with FAO region of Asia Pacific, based in Bangkok, and inspired by the Asia Pacific experiences with headquarters in the Global InFARM initiative. We've added animal species, bacterial species, food types, antimicrobials, the breakpoints. There's also the WHO E. coli Tricycle project, especially with the focus on integrated surveillance and a special focus of water samples, environmental samples. I mentioned the breakpoints. We do give, when people install WHONET, you get one data set with approximately 600 human health isolates. You get a second data set with 400 isolates. Those 400 isolates have 100 human, 100 animal, 100 food from one surveillance project. Those three data sets, human, animal, food, are from one research project. Diseased humans, healthy animals, and food from the market. We then later added 100 isolates of water from an unrelated, completely different country, E. coli Tricycle project. That project, of course, has a lot of ESBL producers. So we do give you a database to play around with, and I'll show that database today. I'm going to go through this very quickly. I'm the co-author of this document from 2011 called VET05. There's a similar one on the human side called M39. It's guidance for analysis and presentation of data. VET05 specifically on bacteria for animal origin. I'm just going to go through this very quickly to just inspire you about the kinds of things you can do with resistance data. Many people only look at their data once a year, which is unfortunate because you're missing issues about quality. People getting, I've seen so many isolates of VRSA, vancomycin-resistant Staph aureus. I'm sure that over 99% of the time it's laboratory mistakes. They're missing outbreaks. They're missing important new things. If you only look at the data once a year, that is an important use, antibiograms, national reports, but you would like to look at daily, weekly, monthly, quarterly, where it could actually impact action. Here on the left, you see imipenem resistant, I think this was E. coli in this example. On the right, these are data from CLAB. CLAB is a commonly used laboratory information system widely used in Africa. It's been introduced also to Cambodia, Laos, piloted in Bangladesh. So those of you who have not heard of CLAB, I recommend looking for it. We're happy to provide links for it. This is just typical percent resistance. I won't go into any detail. This is looking at trends over time. Top left is from the Netherlands, and in dark red, you see that multi-drug resistance has been decreasing in the Netherlands because of all the attention they've paid to growth promotion, banning, and other strategies to decrease antimicrobial use, improving hygiene. And that green color is pan-susceptible, so you see an increase in susceptible. It's top left. Lower right is the lower right-hand corner. I already mentioned the first 5 years of this example. These are salmonella and fluoroquinolones in the United States. And for the first 5 years, I told you it was correct. Salmonella resistance, ciprofloxacin, did not change in the first 5 years significantly. But now if you look from 2017, it's been going up rapidly. And that number, I think it's hard for me to read it, 50% resistant. So the importance of the long-term monitoring of these. These are MIC or dish diffusion data results. Again, these are data from Africa, from CLAB. We download the data, so CLAB has been a wonderful resource, and it's used in maybe 20 countries now in Africa. So I think it'd be a wonderful opportunity for collaborative surveillance and research in Africa, of course, with the buy-in and permission of the countries. So we're having discussions with the CLAB developer and the countries about that. The CLAB developers, it's IZS and Zooprophylactic Institute in Italy and Teramo. Lower left also highlights this issue about, let me highlight on that, clinical breakpoints in the bottom row and then the wild type, non-wild type in the top row. This is what we call a squashed histogram. It's a histogram, except it's in the form of numbers. And it's a convenient way to consolidate a lot of data. Top left, instead of having 3 histograms for cattle, pig, and chicken, it's 3 rows. So it's just a consolidated way to present a lot of data efficiently. You can do a scatter plot to compare 2 similar drugs. People do this all the time with tuberculosis, and they're looking at MDR-TB, but it applies to any combination of bacterial or antifungal agents. Multi-drug resistance, important concept. There are 2 ways to look at MDR. Top left is the ECDC 2012 publication. I'm a co-author of that. I did the data analyses for it, where we propose definitions for multi-drug resistance, extensively drug-resistant bacteria, and pan-drug resistant. And let's see if I can zoom in on this. And this is the NARMS website. I selected bacteria, salmonella. I selected the variable resistance to greater than or equal to 3 classes, chicken, cecal isolates. And you see from 2013 to 2023, a steady rise from 10% to 40% of multi-drug resistance as defined by resistance to 3 or more agents. In contrast, if you look in the lower right, Dutch data, Netherlands data, resistance to 0 classes has been going up. It's been going up, and the purple color, greater than or equal to 3, in the recent years has just been going down, highlighting the impact of various measures, one of which was banning growth promotion. So you can see the tie-in between data and public health policy and food production, very different concerns than what you see on the human side. So multi-drug resistance can be defined by the number of classes. I think that's very valuable from an advocacy perspective. For infection control, molecular typing, understanding resistance, what I find more valuable is tracking the specific antibiotics. These are data from salmonella. Top left, you see that these bacteria are resistant to ASSUT. That's ampicillin, streptomycin, sulfa, and I think that's trimethoprim, not tetra. I think so. That's resistant to those 4 agents. Lower right, ampicillin, the C is chloramphenicol, streptomycin, sulfamethoxazole, trimethoprim, amoxicillin, clavulanic acid, ceftriaxone is that code, and ceftiofur. So this is precisely which agents. So that obviously has implications for genomes, which genes are there, just for outbreak detection. We did a study. I was looking at some data in Cambodia, and we were seeing that from week to week in the local chicken market, the resistance patterns were so different from week to week, but it did reflect that week to week, the trucks come in from different farms, from different parts of the country. So there's no outbreaks. It's just that the source was changing so quickly. It was one market, but the source of chickens depended on which truck happened to arrive at that time. We have outbreak detection. These are also data from Africa from the same group. Top right is from Argentina. It's looking for this is human health data, top right, looking at Shigella sone and using SXT as a marker for a phenotypic marker for the potential genotype for the strain. So we use the resistance pattern. We study multi-drug resistance partially because of its clinical importance, but also it's a free marker. We can't do sequencing for everything yet. So these are strains specifically resistant to trimethoprim sulfamethoxazole. That's human health data in the top right. Lower are Staphylococcus aureus data from animal health. And these are routine diseased animals, meaning I can use this for outbreak monitoring and detection. I would not do this with healthy animals. Why? Because in healthy animals, I might go the first Monday, I might go, May 1st, April 1st, June 1st, and I collect 50 samples every time I go. That's not an outbreak. It's just a reflection of when I go to collect the samples. But for disease data, as long as the disease samples are coming in regularly, you can look for outbreaks. Lower left, it's a Staphylococcus aureus resistant to tetracycline. Lower right, Staphylococcus aureus resistant to ampicillin, kanamycin, streptomycin, and tetracycline. So this is a window into the genotype. Here we have Netherlands that they've linked decreasing use to decreasing resistance. Lower left is profitability. They have demonstrated not only the animal health, the animal welfare aspect, which is one of the statistics here, but also the economic cost. What is the cost of the hygiene and the growth promotion and all these other things? So I want to demonstrate the food is safe, but also the production is economical. And we need to do those kinds of studies in low resource countries to see whether the same conclusion will hold. These are data from my mentor, Tom O'Brien, who passed away a little over a year ago. In the 1980s, published in 1993, looking at genomically, using plasmid typing at that time, RIFLEVs, restriction fraction length polymorphisms. He looked at processed poultry and poultry processors. He always had a way with words. And he found there was very little connection in his study between the plasmids in the chicken and the plasmids in the chicken processors. There were some examples, but in his study, a lot of them did not overlap. That's in contrast to this study. This is from the Netherlands. Alejandro Garcia, Dora Garcia from FAO was the lead author on that. The conclusion in the top right, I'll focus on the top right. It's hard to see the numbers here. 6, 12, 17, 16. 6, the broiler farmers. 12, the chicken meat at slaughterhouse. Those are the broilers. 16 and 17, is that right? Yeah, 16 and 17 were the broilers and the laying hens. So in this particular case, there was a lot of overlap between the broilers and the people taking care of the broilers and the slaughterhouse chickens. The final conclusion of the study is, except in those cases, there's little similarity between ESBL genes in the human and the animal populations, the farm animals, with the exception of people who work on farms. And then there's a lot of overlap. In this particular study, 1/3 of the human SBLs were considered to be of non-human source. Of course, that would reflect their sampling strategy. How many farmers did they select? Study I was involved with from the Philippines. In the long graphs, you see the phenotypes from 2000 to 2017. Here, for a 3-year period, or in this particular example, a 2-year period, 2013, 2014, they did the whole genome sequencing. So we have great detailed results on the genome sequencing for 2 years. But as the background, we have the phenotype data. So phenotype data is complemented. It's much bigger volume, but the genotype data is much richer. So it's great to bring them together. Lower right is Klebsiella pneumonia, and you see three, all of these, most of them we didn't bother coloring in. We highlighted these last three colors, RP, resistance profile 5, resistance profile 6, resistance profile 7, where one of them did not exist in the year 2009. Two of them existed, but were relatively minor, and those three have exploded. So today, over half, in 2017, over half of the Klebsiella were one of these three phenotypes, which in a different slide was correlated with their genotypes. And you have a lot of them where it's one-to-one matching. Other times the phenotype might have 2 or 3 genotypes associated with it. Backlink, I'll show that as a demo. Backlink is our data import tool. Many of you know that already. The lower left, you have Vitek, you have Excel, you have CLab, you have SunQuest, you have these different LISs. And you can't analyze them directly. They're not compatible. But with Backlink, you can get them into standardized WHONET files. And I'll demonstrate that in next, including CLab. I have my own personal background with animals. Lower left is me, and it used to be me anyway. I was 21 in that picture, sitting next to a hippopotamus skull. Lower right is my cousin's farm. My cousins have a dairy farm in Germany. So those are Stelling cows in northwestern Germany. It's the end of the PowerPoint. Look at the time, we're doing well on that. Adam, any questions? Comments?
Yes, there are a few questions now, so I'm not sure if you want to take care of those now or just, at the end.
Let me take a look. Maybe if you could, okay, I'm going to bring up the chat.
Let's see, 748.
Okay, 748. Okay, 748. Barak, thank you. I have a question. Can we use ECOF values for treating the patients? The general answer, I'm not sure whether you mean animal patients or human patients. Generally the typical answer would be no. If clinical breakpoints exist for that animal, for that human, you should use the human clinical breakpoint because that's the whole purpose of it. The clinical breakpoints consider the microbiological elements, but they also consider PK/PD. They consider other evidence about treatment failures. A good example of this was the carbapenems in 2010, when CLSI changed the breakpoints in the middle of the year. They had never done that before. CLSI and UCAS disagreed with what the breakpoint should be, and the Europeans said, we think those are resistant. And CLSI said, no, they do have a resistance mechanism, but for the PK/PD test suggests the patient should respond. And the patients were not responding. There were examples. So they changed the breakpoint in the middle of the year. So if you're talking about humans, if the human clinical breakpoint exists, use the human clinical breakpoint that exists. On the other hand, if the human clinical breakpoint does not exist, the ECOF would be a good place to start. You can tell the clinician, this is not FDA approved, this is not official, we don't have the clinical correlation, but you can say that it's especially value if it's wild type. If it's wild type and it's an approved drug for that therapeutic, obviously don't give a drug that's not going to work. Don't give penicillin for an E. coli infection. But as long as it's a drug that makes sense and it's wild type, then yes, S should be reliable. The drug should work. If it's non-wild type, that means there's some resistance from a phenotypic perspective. The drug may or may not work. So I would only use the ECOF in the human setting if a human breakpoint did not exist. And I would give a caveat, this is not an official breakpoint, so it's sort of off-label, informal use. The animal study, unfortunately, this happens a ton. There are only clinical breakpoints. If there is no animal-specific clinical breakpoint, you really have just two choices. You're going to use the human breakpoint and label as the human breakpoint, or the ECOF. So in the animal population, I do not work practically in this, so I don't want to give you a definitive answer. I'm just saying it would make sense to me that you use the human breakpoint because that's what exists to a large degree, and not all ECOFs exist. So if there is no animal-specific breakpoint, use the human breakpoint, or the ECOF would also be reasonable, especially if it is a susceptible strain, a wild-type strain. Wild-type, the drug should work. Okay, I hope that answers. Adam, next. I'm looking at 749. Why are there no specific breakpoints where environmental samples are analyzed to cater for both human and environment? Well, so when you talk about environmental samples, you're interested mostly in one of two things. does the bacteria have a resistance gene? And if the bacteria has a resistance gene, then the ECOF, then if that's your concern, then the ECOF would be the best way to find it from a phenotypic perspective. There are other things, not only whole genome sequencing of individual isolates, you can also do metagenomics and look at DNA. You don't have to look at one strain at a time. So there are other mechanisms for looking for resistance genes in environmental samples. If you're collecting water samples to assess the potential impact on human disease, then using the human breakpoint would make sense. On the other hand, if you were collecting water samples to try to estimate the impact on the health of cats and dogs, because we do have those breakpoints, that would be reasonable, which wouldn't be a common use. So I'm hoping with the subsequent comment that I gave, some of the considerations, you can make a reasonable decision for your particular scenario. We also do have experts in this field, so if I cannot give an adequate answer, then I'd be happy to try to find some more official answers from people who work in laboratory or clinical medicine in the animal sector. So yeah, so in the environmental sector, you're either looking for resistance genes, so the ECOS makes perfect sense, or you're trying to look at the impact of contaminated sewage on the human population or the animal population. And if you have a particular animal species in mind, you can use those specific clinical breakpoints. Adam, next question.
I think it's just the next few messages here. We're almost towards the end of the chat.
Great. 756. John, could you share the name of the Vietnamese Integrated Surveillance Program? VNARIS is the human health sector. I don't know the name of the animal health sector. So it's VNARIS. It must be like Vietnamese National Resistance Surveillance, something like that. If you're interested in the animal health sector, I do know those people. I just don't know the name of the network. Someone asked, can you have the recording? And Adam answered that. Or yeah, the answer is yes. We'll make the presentation available. Yes, Adam?
753.
753. WHONET software, no provide alerts for duplication. I'll come back to that. WHONET is not web-based. WHONET does have an entry field for patients aged less than 1 day. No comments, antibiotic therapy, no patient's clinical history entered in WHONET, no capacity indicators entry. And why WHONET software not restrict unnecessary inputs from Ethiopia? There's a lot of questions. I'll just touch on a few of them. For duplication, question about one thing WHONET doesn't do, if you type exactly the same record twice, the same exact details, no, WHONET does not give an alert of that. It's not a common scenario. It's not common. Somebody will type exactly the same thing twice. It is common a patient might have more than one bacteria in one sample. It's possible a patient may have E. coli multiple times. You should enter all the E. coli because the first one might be susceptible, the last one might be resistant. So it is true, WHONET, it's a suggestion. We can note that, but it hasn't been a priority. No one's really requested that because it's rare someone would type exactly the same thing twice. Also, an increasing number of people don't use WHONET for data entry. They use backlink and they download the data. So when you're downloading the data, it's up to the original system to avoid duplication. So your comment is correct, and it has not been identified by users as a priority. It's something doable, but it's not something that's come up. WHONET web-based. We made a lot of progress on a web-based version of WHONET data entry. We started on a WHONET web-based data submission portal for national coordinators, but we had 2 programmers. Now we have funds for 1 programmer, so it's just not realistic. We are having discussions with the International Vaccine Institute in Korea and the Oxford Big Data Institute on a direction for trying to get a web-based version of WHONET. We'd love to do that, especially for a data visualization tool for national reporting. Web-based data entry is less needed, but it could be used because people usually have their own LASs and they use backlink. But if we're doing a research study with like 40 labs, small data volume, we had 1 project with pertussis, 1 project with gonorrhea, small data volumes, and a web-based solution would have been very useful for that. We agree with you, and if somebody has some funding for us to get more programming time and collaborations, that'd be excellent. Patients aged less than 1 day. I'm not sure. WHONET does allow you to put 0 days. You can put 0 days or 1 day. Well, no, I guess if you put 0 days, WHONET probably changes it to a 0. It hasn't been expressed as a priority for ages less than 1 day. WHONET does days, weeks, months, and years. Antibiotic therapy, WHONET is an optional field. Yes, you can go to data fields. I'll show that when I get to the demonstration. You can enter the antibiotic therapy. It's often incomplete. It's often difficult to analyze, but it is possible in research projects. In research projects, people do take the time and effort. Because a lot of times the lab doesn't know what the therapy is. You know, the doctor collects the sample on Monday when the patient gets to the ER. They collect the sample before they started antibiotics, or maybe they change antibiotics in day 2 or 3. It also becomes complex if there are multiple antibiotics and they change the antibiotics. So WHONET does allow you to enter the therapy as an optional additional field. Clinical history, yeah, I can show in Argentina does ask about risk factors, underlying conditions, diagnosis. These are optional additional fields that you can enter. I'll show that. Capacity indicators, I'm not sure what capacity means in this context. And why not restrict unnecessary inputs? Well, unnecessary inputs depends on the user. You know, what is considered necessary, unnecessary. So we try not to restrict people in that. We've never made any required fields or any, you know, and because sometimes people have good reasons that don't make sense to me, but it makes sense to them. So some of these would be valuable for potential development, but we have a list of things and priorities. And when more people say, then it becomes a priority. A bigger priority. Adam, next question, or none more?
Let's see. I think 8:15.
Okay, 8:15. If here's an aquatic animal pathogen of zoonotic significance, would the breakpoint be the same for treating the human fish? Well, for example, a lot of them don't have a lot of I'm not aware of any, I'm not aware, it doesn't mean they don't exist. I'm not aware of any VetCast, UCAST aquatic breakpoints. CLSI has the VET03, VET04 document, and they have therapeutic breakpoints for diseased fish. So that's the purpose of those. There's Aeromonas hominosida, I don't remember what else is there, which is actually not on the human pathogen list. So it's different from treating a human. So you would not, you know, I don't think in humans we have a breakpoint for Aeromonas salmonacida. I think it would just go into, I think Aeromonas is a non-fermenter. It might go into the default non-fermenter group. So I would not simply take the fish breakpoint for treating diseased fish and apply it to humans. You might do the ECOF. Keep in mind if it's watery diarrhea that the human gets, a lot of times the treatment is not antibiotics. A lot of times the treatment would simply be fluid electrolyte replacement. I'm not an expert in that, but my first thought is you don't take fish clinical breakpoints and use them outside of the fish that are indicated there. It is conceivable that the VET03, VET04 might have some. CLSI does have a few ECVs, a few ECOFs. Most of them are clinical, but they do have some ECOFs when there's not sufficient clinical data. I want to know about antibiogram, which is not including all organisms in which is selected. You can select, when you do an antibiogram, you can select precisely which organisms you want. So I think I'm going to finish the questions and get to the software demonstrations. So let me share my screen. Okay, so I'm going to click on New Lab again. Let me call this United States. Let me call this One Health webinar. And OHW, Human Animal Food Environment. So antibiotics, not much to show here. You know, basically, just like always, you choose ampicillin. And then I'm going to say ampicillin CLSI, and I select OK. I can also do enrofloxacin. So this is an example of a veterinary agent. Tetracycline is both human and animal. I'm going to go to breakpoints. So right now you see at the bottom, it says use data-driven breakpoints. That is the new way I was mentioning. This is the old way to use they're saved in your local configuration. Data-driven, it just reads it live from the WHONET tables. So that's how you change from the old way to the new way. You know, of course, as I mentioned, WHONET will ask you, and if you say no, if you say yes, it'll switch to the new way automatically. If you say no, you can switch to the new way here simply by clicking on user-driven breakpoints. You can configure that, and you can say, I want the human, I want the animal, I want the food. So you say which breakpoints you want. Adam, I thought all of those three would be checked off. Let's just double check on that. Maybe I did not check on the other screen. And then here at the bottom, you see the breakoff, you see human here, you see ECOF here, animal for swine. So the breakpoints are all here. So if I go to breakpoint type, these are animal breakpoints for swine, cats, poultry, dogs. It's especially cats and dogs, but a few others. If I go to ECOFs, these are all the ECOFs. So they're all in there. Let me click on cancel. I'm not going to change any of those. Well, actually, I do want to, did I click on, no, Adam, just, if you could just double check that later. I've checked here on all four, but when I went to antibiotic breakpoints and I clicked on configuration, I don't know whether I need to check this off. So if you could just double check that later.
I think it was just a decision we made at one point that we weren't going to enable those.
Okay,
We can,
right.
we'll revisit that.
We'll revisit. The way you remember, I'm sure, is the correct way. Okay, good. I was the programmer for WHONET until around 2012, and then I did a little bit for a few more years. Since then, Adam's been doing all the programming. Okay, breakpoints, and so you see the breakpoints are there automatically. Nothing else to show here. Let me click on OK. Locations. I'll show you what I would do for a human example. For a human example, I might say something like neurology ward, neuro, hospital OHW, medicine inpatient. But if I want to do an animal location, I can do Farmer Brown, blank, not relevant, farm. I can say restaurant, McDonald's number 5, RES restaurant. I can say, for example, Nile River site number 10, Nile 10, and I can call that environmental. So the two middle columns are only relevant for human health. The other columns you see down here, cafeteria, food, animal, ICU. Adam, I would like to make a change here so we could filter between human and animal food so people can easily find the animal stuff together.
Okay.
I've thought of it before and I keep on forgetting on it. Because right now everything is mixed together. I see restaurants, slaughterhouse, veterinary clinic, but also I see inpatient, ICU. So let's just, let's keep it this way by default, but we can also offer a drop-down option. So for defining locations, the two columns on the left are up to you. They define the precise name and code of your location where the samples were taken from. The column on the right is just to introduce some degree of minimum standardization. These are the farms, these are the veterinary clinics, these are the water collection sites, these are the restaurants, and that's more useful for FAO, national data managers, sharing data between laboratories where at least some things are different, but other things are the same. Okay. Data fields. I go to data fields. If I said only human, you would only see human health questions here. The traditional questions appear at the top, first name, last name, sex, etc., date of birth. And when I click on animal, at the bottom, it should say animal species, animal type, for example, the production type. Is it a meat animal, an egg animal, a work animal? Things like region, county, some of those broader geographic, farm number. If I go to the top here, if I click on last name, that's a human question. We don't ask for the other sectors. First name, it's a human question. I'm joking a little bit that we don't want to know the animal's name, but actually I'm just joking a little bit because if you work with cats and dogs and horses, people often do keep track of the animal's name. You know, the animals on the farm don't have names, but the animals at home and the horses do get names. So I have a veterinary system where they do keep track of the companion and large animal names. So it is configurable. Date of birth, obviously animals have dates of birth, but we don't usually know those dates of birth. But you can say yes or no as to what fields you want to include or exclude. And then here, if I click on modify laboratory, modify list, I can change the list. For example, if I only work in outside of the human health sector. So first of all, as I said on the previous screen, if I enter food, it is not going to ask me first and last name because they're not checked off. The field is still there, but it doesn't show up on the data entry screen for food or animals. But I can go a step further. I can remove the question. I don't want the last name. I don't want the first name. I don't want the date of birth. If it's food, I don't want to think about the age or the gender. So you can remove questions that you don't need. If I go to veterinary, let me go to FAO InFARM. FAO. FAO, you see 3 here from the region of Asia Pacific: diseased land animals, terrestrial, diseased aquatic animals, and healthy animals. Those 3 modules came from region of Asia Pacific, from Bangkok. And then FAO InFARM came from headquarters in Italy. So FAO InFARM, they ask data surveillance program, are these healthy or diseased? Are these land animals or aquatic animals? What is the animal species? What is the animal type? What is the market category? Optional, what is the latitude, longitude? We don't usually do that in human medicine. I mean, you can if you have the patient's home address, but it's much more interesting in the environmental setting or maybe farms where there's so much outreach. Humans, they usually come to you, whereas the food and the animals used in the environmental sites, we go out to there and you can do geolocation. The most important fields we put at the top: animal species, surveillance program, data year. Why do we ask data year? Because many times we don't actually know the specimen collection date. Because the samples are collected on the farm on Monday, they might not get to the lab until Wednesday. They go on a truck and they eventually get to the laboratory. And many times they do not record the collection date, they record the receipt date. So the FAO just wanted something they could rely on. So the data year is something they could rely on. If I go to NormVet, was the human sector in Norway. NormVet, I don't think they still utilize this, I'm not certain, but they would ask about mastitis, bovine mastitis, lactation day, lactation type of inflammation, age of mother, anatomical site. If I go to Vietnam Animal Health, they were asking breed, sampled food, veterinarian ID, region, province. They want to know where's the animal from. They want to know where's the sample from in case the sample is in a different city or a different district, collected by. So it's a long list that they agreed on. So as someone said earlier about what is the clinical diagnosis, what is the treatment? Yes, you can add these. Either find it on the WHONET list, or if you don't find it on the WHONET list, then just click here, user defined, select, and then I can say, I don't know, outcome. Outcome's not a good example because actually outcome is one of the WHONET options. Let me just put, for example, PCR ESBL, something like that. PCR ESBL result. Text field, 10 characters long, okay. And that field now appears at the bottom. Adam, small thing, if we had a new field, if we could just highlight that and just make that the active thing, because otherwise I just have to scroll down to see it. Okay, and so that's the PCR. You can also put the name, like for example, veterinarian ID is already on the list, okay. If I go to veterinary ID, I didn't select the field. Let me do that again. If I go down to Vietnam again, Vietnam, animal health, and I go to veterinarian ID, I'll select that. It's now at the bottom of the list. So now veterinarian is here at the bottom of the list. I have the ability to go to code list. And I can make a list. No code validation, I'll just take any veterinarian's name that I give. Or I can type in the names of the veterinarians here, or the names of the districts, or the cities, or the provinces, or the PCR results, positive, negative. Or I can link to an external file, and then you could keep the file in Excel or Access or something else. So WHONET does allow you to add additional fields, as well as additional drop-down list. I have talked about this previously when I talked about WHONET Modified Lab, and on the human side, some people use it, and it's very valuable. On the animal health side and the research side, people use this very often. You know, in Colombia, they wanted the list of their regions and the subregions. So this is a feature that exists for humans and animals, but they're often much more commonly used. So often the non-traditional hospital use are the people who really push a lot of these WHONET features in a good way, because we try to make it very flexible. Your veterinary ID is at the bottom, and that is a human animal food, not environmental, but you can say yes or no, and you can check things off. And then I can do move up, move down, so that you can put it, you can put the question wherever you want. I click on OK. I click on save. I go to data entry. I say new data file, and I'm now in the new data file. And now you see veterinarian ID is right at the top where I wanted it at. And on the lower right here, you don't see a list of veterinarians because I didn't make a list. I didn't make a list, so I just type Smith and accept Smith. If I do make a list and I put Smith and Smith is on the list, great, or I can click on Smith. On the other hand, if I type Jones, if Jones is not on the list, not a problem. WHONET simply says, Jones is not on the list, and it still accepts it. WHONET warns you this is a new person, a new whatever. We just want to warn you that it might be a typing mistake, but you might say it's fine. It is correct. I just don't have the time to add it to the list, or I don't. If it's a veterinarian who comes every week, yes, let me add him to the list. If it's a veterinarian that comes once a year, I don't have to add everybody to the list. Adam, just a thought I've had before, but I never think to tell you these things. We do tell the person this is not on the list. It would be a nice feature if we could ask them, would you like to add it to the list? That would save them having to go back to the configuration area, and then it could just be added here automatically. Okay. So here I'm under location. You do see those 4 locations: Farmer Brown, McDonald's, neurology. And when I click on it, it automatically says neurology inpatient, or Farmer Brown is a farm. So those first 2 boxes about the animal and the location, those are very customized for human animal food environment. The other question, specimen, microbiology, that's pretty much the same across the sectors. Here at the top, I've selected animal. If I select human, the questions at the top are different. If I select food, the questions are different. Under food, we do provide, here on the right is a list of codex. Codex is an international standard for many things related to food production, food safety. One of the things they make is a list. For example, if I see here eggs, there are different kinds of egg. There's 10.0 eggs, then 10.1 fresh egg product, preserved egg or egg-based dessert. Adam, that's misspelled. It should be dessert with 2 S's. Funny, I would just choose that one. I'm getting sun in my eye. I'm going to move my table a little bit. Okay. If I go to environment, we have a lot of people obviously using human on the human side. An increasing number, a good number on the animal health side, a small number on the food side, and very, very little on the environmental side, except as it relates to the new E. coli Tricycle project or to the old WHO Agisar projects. So if any of you find that WHONET is good, but it could be better, you could send us recommendations. Please add these data fields. Please add these drop-down lists. We might add them as a standard default that everybody gets, or we might put them on that optional list that I showed you. That we have the Norway, we have Ireland, we have the Vietnam. So we do appreciate, because WHONET is only as good as, I'm not the expert in these things, you're the experts. So if you give us guidance, some of your requests are very easy to do. Some of them are really not feasible to do easily, quickly, and some of them are worthwhile, and we try to prioritize and we try to get to them depending on how important it is, how many people would benefit from it, and how much work it would be. So environmental, we have not yet added, what we did add, and then we removed it, agriculture plants and animal feed, because I said we just don't have guidance. I don't know what list of plants to put. Corn, because they have their problems of resistance and antibiotic use. You have food plants, apples and oranges, and then you have orchids and other commercial flowers. You have cotton. So we don't have enough expertise to put that on the list. But you do hear more about it in these group discussions. Okay, certainly not. Okay, let me start with human. If I say Streptococcus, okay, I'm going to exit out of data entry. I'm going to open up a real laboratory, the WHO test laboratory. I'm going to click on open. I just want to highlight some features. I'm on human. I'm going to put Streptococcus pneumonia. And if I go to e-test, penicillin, and I put in MIC of 1, you see here that there are 3 interpretations, SRI. S are the non-meningitis breakpoints. Meningitis breakpoints and oral breakpoints, that's especially when using penicillin for oral therapy. So CLSI has 3 different breakpoints. UCAS has 2 breakpoints for that. If I go to ceftriaxone, there are 2 breakpoints, meningitis, non-meningitis. Let me go to something like, let me change at the top left, I'm changing human to animal. Let me change the organism to E. coli. Let me put ampicillin 20. It says SSS. The human breakpoints are 14 to 16. The cat breakpoints for uncomplicated UTIs is sensitive greater than or equal to 17, and same for the dogs. In fact, that agrees with the human, but the human sensitive starts at 17. So for human cats and dogs, sensitive is 17. They don't agree at, for example, 15 millimeters. For 15 millimeters, the human is intermediate, and the animal is non-susceptible. Non-susceptible is slightly different from resistant. Susceptible means the animal should get better. Non-susceptible, it's not exactly the same. It's quite similar to the idea of Eckhoff. It means it doesn't look phenotypically susceptible, but we don't know whether it's clinically resistant or not. So in this case, for the human, it's RIS. In this particular example with cats and dogs, there is an S breakpoint, but there is no R breakpoint. So it's either susceptible or it is non-susceptible-ish. So that's why it says NS. So you can see WHONET does all 3 breakpoints at the same time. On the other hand, if I say human, and if I go to animal, it does not show me the animal breakpoints. I think that's all I want to show on data entry. Let me go to data analysis, looking at time. We're still good? Good. I'm now in data analysis. Analysis type. Let me just start at the basic place. Let me just do a simple, there's %R detailed. I'll show you the detailed one. And I'm going to click on OK. Oops, I clicked on the wrong thing. Click on OK. The sun's in my eyes, so it's a little bit hard to see my screen. Okay. I'm going to click on organisms, and I'm going to put something simple like E. coli. Okay. Data files. The first file here is January 2020. That is 1 month of human health data. I use that when I teach a human health audience. This database has 400 isolates, 100 human, animal, food, environmental. As I said earlier, human, animal, food is mostly E. coli and Salmonella from one research project. It was funded by Agisar, WHO. And the other is from an environmental E. coli Tricycle project. So I just threw in 100 E. coli and just changed the dates so it all sort of matches. So I'm going to choose that one file. Of course, you can choose as many files as you want. You say, OK. Begin analysis. And it gives me my detailed report. I like to start here on the graph number tested. And here I can see the antibiotics that were tested and how often they were tested. Here at the top left, you see ampicillin 3 times. I'll explain why in a moment. Ampicillin is always tested. Nalidic acid, Cipro, always tested. Trimethoprim sulfa, always tested. TZP is piperacillin tazobactam, infrequently tested. imipenem, sulfoxygen, infrequently tested. So this allows me to see what drugs they are always testing, sometimes testing, rarely testing. Keep in mind, I've merged together two different databases. One, it was human animal food from one project with the same set of antibiotics, and then a different project. So some of the antibiotics overlap between the projects. Okay, why is ampicillin there 3 times? And you see why in the table. It's once there with the human, cat, and dog breakpoints, and ECOFs optionally. So you can also have the ECOFs here. And if I come over here, you can see that, yeah, E. coli is mostly resistant. So if I look at the human breakpoint, so this is, yeah, okay. So here are 3 histograms. This is ampicillin using the human breakpoint. This is ampicillin using the cat breakpoint and the dog breakpoint. So as you can see, the graphs are the same. The only difference is that the human breakpoint has an R vertical red line, and the cats and dogs don't. In fact, the cats and dogs, they agree that it's the same breakpoint as the human. Then, yeah, more examples like that. That's enough of that, and you can see site of infection, horses, adults, breakpoints. So I think in that case, there are no, that's right, in that one, there are breakpoints for adults, but not for child horses. Gentamicin, maybe the drug is not recommended, maybe because of their kidney. I don't know what the reason is, but obviously CLSI felt there was an important reason. So that's presenting the data, but this is human animal food all averaged together. How can I change that? Let me go to continue. I go to analysis type, and here it says row number 1 is the antibiotic, of course. You see 1 antibiotic on each row. But I would like to stratify it by origin. I'm going to click on OK. I'm going to click on Begin Analysis. It's now going to be the same data, but now you can see ampicillin. Well, let me choose a simpler one without, let me choose a simpler, something without all those breakpoints. Okay, so here you see sulfoxygen 3 times. Here in this column, it says A. Those are the sulfoxygen results for the animal isolates. So if I go over to the right, there were 6 animal isolates. There were 10 food isolates, and there was 1 human isolate. So I can see the 3 different sectors, and apparently there were no environmental tested with sulfoxygen. So I can see all the results together stratified, as I showed in the PowerPoint, where I just had cattle, swine, etc. on different rows. Here I chose the sector as the variable. I could have been animal species. So this could have been 1 row for chicken, 1 row for cattle, 1 row for fish, 1 row for human. So it's one way to stratify. I'm going to do continue. That's the detailed report. I'm now going to go to organisms. But no, I'm not. I'm going to go to RAS analysis type. And I'm going to go to the summary. This is what we call the antibiogram report. I'm going to click on OK. Oh, and same thing. I would like to do the rows as origin. We may or may not change the word origin because the word's confusing. We might change it to sector. We might leave it at origin. I haven't decided. But when I say origin, it means human, animal, food, environmental. The problem is we started doing a lot of this before other groups joined, so we had to make these decisions on our own before we had a lot of people to tap into, because even at that time, the people were using different words. A change I've mentioned on a previous call when I talked about antibiograms, previously WHONET allowed you to choose %S or I or R. Now you can choose more than one, and that becomes particularly important once you want to keep track of the susceptible to dependence and the ECOFs and the wild types and the non-wild types. So you can check them off. I just want to do the % susceptible here. And let me do OK. Let me do begin. And now what you see are 4 rows. You have all of the animal results on row number 1, 24 isolates. The environmental row, 100 isolates, as I told you earlier, on the second row. 10 and 19 food isolates, 25 human isolates. So this is what I call data stratification. Just stratify it, just choose origin or animal species or farm or region, and it will show you the antibiogram for each value for the variable that you have selected. On this example, let's see if I see anything interesting. Before I do that, where's imipenem? Oh, not a good example. There's nalidixic acid. I want to scroll over to the right. Here on the right, you see the denominators. You see cefoxitin was only tested 5 times. On this last one, cefepime was only tested once. So imipenem was only tested 5 times here. So keep in mind that these numbers on the left don't tell the full story. Some of these numbers only represent 1 or 2 isolates. I'm going to do continue. This is a feature I have mentioned on a previous call when I talked about antibiograms. I'm going to click on analysis type, minimum number of isolates. The CLSI recommendation is 30. We drop it down to 20. That way you can go up to 30 if you want. But if there are 28 isolates, a bit shame to lose it completely. So we show you by default 20, and then you can decide before you publish what threshold you want to use, and then you can delete any rows you don't want them to see. So now asking for a minimum of 20 isolates. So many of the antibiotics I just showed you have just disappeared because it did not meet the minimum. And now when I scroll over to the right, all of these are at least 20 isolates. Let me do continue. Okay, that's one thing I wanted to show you. To do by sector. I'm now going to go to isolates. Go by isolates. Origin. What I showed you were human, animal, food, environment stratified by different rows. But maybe I only want human, or only food, or only chicken, or only cattle. So I can go to origin, and I say I only want the animal isolates, or the animal and the food isolates. I can include or I can exclude. I will do include. Okay, begin analysis. And apparently it only gave me animal because there were not a sufficient number of food isolates, for what I requested. I'm going to click on continue. I'm going to stop sharing. Well, I'm not going to stop sharing because I'm a little bit afraid of freezing the screen. Look at the time. Adam, are there questions?
There were a few that have come in since you finished last.
Okay, before getting to the questions, I'm going to go to the internet, and I'm going to go to UCAST. And I say accept everything. I never know what I'm always nervous. UCAST has dramatically changed this front page. So I don't even know where they put it. Adam, do you know where the veterinary stuff is? It used to be on the left-hand menu. Let me put breakpoint tables. It's probably under breakpoint tables.
It might be at the top there. You see VetCast at the top.
Oh, I see. The top menu, VetCast. Thank you, Adam. Yep. VetCast. VetCast overview. I need to review this again. And you see here the ECOF values, development of clinical. So they give guidance on how they are doing clinical breakpoints. But the only one I remember they had was Tylosin. I forget which one it was. I need to ask the VetCast people, where are the breakpoints? Because they do get presented, but I haven't looked at the website recently. Maybe they're there now. If I go to MIC distributions, and I can click on bacteria, and I'm disoriented because I haven't, here's lower right, MIC distributions, view MIC UCAST, allow all, and search database. Okay, this screen I know. So I can choose, for example, ampicillin. And here you can see ampicillin breakpoints. This is the MIC data. And on the right, you see the ECOF or the TECOF, the tentative versus the confirmed. The TECOF is 0.5. ID, I think, means in development. So this is where you find the MIC distributions in the main table and the ECOF off to the right. Or if I choose species, and if I choose Acinetobacter baumannii, you see all of the breakpoints there. That's MIC. If I click on diffusion, you see the wild type appear in black. And the non-wild, so for if I look over to the right, you see that the ECOF is 18. And you see 18 is the start of the wild type area. 17 is non-wild type. So that's how you read this table. Okay, let me go to CLSI Free. And they have also reorganized their website. It's in here somewhere. Access CLSI Micro Free. And submit for the full submit. For lack of time, just continue, and you can get the human M100, M45. You cannot download them, you cannot print them, you cannot copy paste them, but you can read them. M100, M45 on the human side, and a few other methods documents, and the VET01 on the animal side. That's routine bacteriology. You cannot do the VET0304 for aquatic, and you cannot do the VET05, which is fastidious and rare organisms. It's there. They reorganized the website. I'm not going to show that. I totally agree with Gabriel. There should be an alarm for duplicates or a way to choose how strict or lenient it is. I've seen cases where the same patient ID, lab ID, age, specimen number, specimen date, but two different genders. Okay, Adam, let's note this down, because we do note recommendations, and then we have to consider how much work it is and how many people would utilize it, et cetera. Thank you for that recommendation, because that is how we prioritize our programming work. We'd love to have a second or third programmer, because right now we do have to juggle a lot. Adam, the next one?
Let's just move right down. I think it would be 826.
Yeah, is it possible to implement internal laboratory quality control and data analysis for microbiology labs? There is a QC range file. Is it possible to set these up? Oh, that happens automatically. And yeah, sure. I'm still sharing my screen. I'm still in WHONET. I'm going to go back to data entry. If I type, it's not obvious is the problem. It actually does say it here in the lower right-hand corner. It does give an example. So if I say ATCC 25922, any microbiologist in the room, I hope, knows that this is an E. coli. Automatically, the specimen type is QC. That's an E. coli. And then if I type, for example, 10, 20, let's type that, 20, 30. The QC range, so amikacin, the proper range is 19 to 25, or valid values. 10 is too small. So you get the comment somewhere, the result is out of QC range. So yes, the QC, UCAST, bacteria, fungal, breakpoint, QC ranges are all there. Okay, next question. Is it possible? Okay, the next question. I was thinking it'd be good to increase the number of preventing duplication options. Furthermore, it would be beneficial if it were made easier to assess combined antibiotic resistance, imipenem for carbapenem resistance. So WHONET does allow things like that. For example, you can say, show me isolates that are resistant to imipenem or meropenem or ertapenem. So WHONET does have features that can do that. WHONET has a lot of features that can do that. And we can, if you write to WISH, we can give you guidance. There's one thing that we do not yet do that we would like to do, but it's just more time and priority setting, is it's a bit annoying as a national data manager that some of the labs do ceftriaxone and some of the labs do cefotaxime. WHONET, there's not an automatic way in an antibiogram to connect them together. So you have one column with the cefotaxime statistics, a different column with the ceftriaxone statistics. So in answer to your question, WHONET offers a lot of features to do exactly what you described, but it's not complete. I would like the ability of a national coordinator to group together the fluoroquinolones, to group together the carbapenems for the ease of the end user. So we've done a lot already for that. WHONET has the ECDC, MDR, XDR, PDR definitions. That's automatic. We have the resistance profiles, number of classes. So WHONET can do a lot like that. And there was actually a webinar, I don't know, a year and a half ago on multi-drug resistance features. So I might direct you towards, go to the WHONET webinar website and look for the webinar on multi-drug resistance. But it's not complete. I would like to have that ability to merge together, similar drugs. Okay, can the software be updated such that when data is lost, it can be retrieved and restored? The problem is it's like Word or an Excel. It's your job to make sure things are backed up. And so WHONET does have actually we can maybe Adam, you might want to make some comments on this or not, depending on what I say. We do have a WHONET automation tool. And the WHONET automation tool does allow automated backups and copies, an automated backlink, and automated WHONET, and automated emails. So we've already made the WHONET automation tool to allow these things. But don't think of WHONET as like extremely different from Word or Excel. You need a strategy. One strategy is manually back up your files once a month or before your next big trip. Another one, a better one, is like automated daily. Like our hospital network, they back up stuff all day long, 24 hours a day. I don't know exactly what their strategy is. One good strategy is don't save the data on the local hard drive. Save the data on a network drive, because the hospital network, laboratory network people are usually pretty good about doing backups, at least weekly, at least monthly, potentially daily. But when people say we lost our data, sometimes they didn't lose the data, but they don't know where the data are. Or they're on some location that they don't have the password, or the server is down, or it's just in some other folder. You know, you have a new data manager. This data manager kept the data here, and the other data manager is looking over here. So sometimes this is a lucky situation. Sometimes when people say the data are lost, they're not lost. They're just someplace else. But you know, if you lose a Word file, Excel file, there's no automatic way to get it back. So we highly recommend proactively think about backing up your data, and storing the data on a network drive is a very good way to do that. Some people store their data on Google Drive or Dropbox. In my hospital, we are not permitted to use normal Dropbox for anything confidential, but we do have a contract with Dropbox, and we have Dropbox for Business. So we are allowed to save confidential data through a business associate agreement. So, you know, when people lose the data, it's like if it's a virus, it's a computer damage, you know, it's a loss, it's a theft, it's very little that we can do. We can help you to find the data. Sometimes we can get the data back, but please think about proactively having a backup, because if you lose your Word file, you know, losing a WHONET file is not very different from that. Slides will be available. Can we edit backlink data fields? For example, can we add urology department to department fields? Adding urology department is a feature that WHONET offers to add it to the list. I'll show what you a backlink does if I can get to that screen easily. Backlink, you cannot change the backlink list, but you can enter free text. So the answer to your question is sort of yes. I did some work recently with one of our countries for chicken thigh food data, fish, livestock data. That's just why that's on the screen. If I go to new, and if I go to junk, and I say, I'll just do something quick and easy, Vitek and Vitek data, and I say, okay. And I say, oops, I said the wrong thing. And I go to save, and I go save, and I go exit. Junk, browse, and I choose my Vitek data file. Say, okay, conversion. Okay, good. So you see here it says ICU. Here it says 5 North. Here it says 3 South. Do you want to review the new codes? Yes, I do. And location, define codes. And I'll go to 5 North, and I'll say define code. This is a very specific question, so if you're not following this, don't worry about it. So here in WHONET, you can add urology department to the list. In fact, you cannot add to the list. You could call it surgery if you want. It is part of surgery. But you can also use this. Let me just put this back to blank. You can type Euro here. So WHONET backlink does allow you to enter free text. And I'll say that's an inpatient. So I'll say OK. And then if I look at the dictionary, you'll see it's been defined as Euro. So no, you cannot add urology in backlink to the list. But yes, you can add it to the code mapping. So yes, you can do what you have just described. Vitek, all combination drugs, like pip/tazo, ceftriaxone, what to do. Oh, but there's no problem at all. Pip/tazo is, as far as Vitek is concerned, is one drug. It's a combined drug, so you don't have to do anything special. I'm going to make a comment about trimethoprim sulfa, but all the other drugs, all of the IT systems ignore the concentration of tazobactam and sulbactam and avibactam. So it's just that the MIC is the MIC of the piperacillin. It's the main component. With trimethoprim sulfa, with clavulanic acid, tazobactam, these are not antibiotics to a large degree. They're helper compounds that are inhibitors that permit the antibiotic portion to work. Trimethoprim himself is different. It is two different antibiotics that work synergistically. So almost all systems will either just put the trimethoprim portion, 1, 2, 4, 8, 16, or the Vitek adds the two numbers up. So it's 1 microgram of trimethoprim and 19 micrograms of sulfamethoxazole. So it's a total of 20 micrograms. So backlink automatically restandardize it to the trimethoprim. So you don't need to do anything special. If it's called amoxicillin clavulanic acid for you, just match it to amoxicillin clavulanic acid for WHONET. No other special thing is needed. And also if it's a Vitek, backlink is going to understand automatically. Here I'm under junk Vitek, and I go to edit format. I showed you in WHONET how to add and remove data fields. You can do the exact same thing with backlink, very valuable for food sector. I'm going to click on file structure, data fields at the bottom, modify the list of data fields at the bottom, modify, and this is the list, standard list, and you see all of the Vitek things like Vitek is bio number, Vitek probability, Vitek testing date. So you have the normal fields, WHONET fields, plus the additional ones. If I go to modify list, and if I go to FAO diseased animals, these are the questions, aquatic animals, but InFARM, these are the questions. So FAO would like people to add these 3 fields at least. It's the minimum required. Now they're at the bottom. Surveillance program means healthy or diseased, land or aquatic or food. Data representation means systematic, national, pilot, local. So it's just trying to give a sense. Is this a national program or a local program? And data year, simply the year of the data, knowing that the lab does not always know the precise date of collection or receipt. So just like I showed you for WHONET, backlink has the same thing to add or remove things so that this field, this list of fields that you see here can basically match your Excel file. Okay, save. I'm trying to think if there are other things I wanted to cover. Is WHO operable with DHIS2? That's a complicated question. We've done, it's a simple question. The answer is complicated. We have done a lot of work. It's on the WHONET screen, and we do have guidance on this. We made a lot of progress, and then we stopped on it, but I'd love to get back to it. MSF is interested. We did it for some of the Fleming Fund work, and MSF has some interest that we're trying to learn more about. If I go to, open laboratory, data analysis, quick analysis, there's an option here called DHIS2, and there's a document for how to utilize that. And we have 3 examples there. That's one place. Or if we go to data entry and export, Adam, I don't quite remember which of these options. Oh, here it is. It's just there listed DHIS2. So it's also here as a DHIS2 export option. And then we have another option on the data analysis screen, DHIS2. So yes, we have done a lot of work on it, but we'd like it to be optimized. One thing we have encountered, for example, Vietnam highly recommended DHIS2 to me several years ago, and then they gave up after 2 years. They said it's a lot of custom programming, the performance is too slow, and we don't know that many people who know DHIS2. So then they just hired college graduates in computer science, and they ended up with a very traditional and excellent, web portal using traditional tools. So we've worked with a number of DHIS2 projects for antimicrobial resistance, and there have been different levels of success, but there are challenges in DHIS2 with the built-in functionality, which is often not sufficient, like first isolate per patient and breakpoints. So you have to do a lot of custom programming, which can slow things down, and a lot of the performance is often not good in the Vietnam example. And you don't have that many people with that knowledge. I do know MSF is happy with their DHIS2 work. We just want to see how we can integrate in that. So we'll learn more about that. So we certainly are very interested in DHIS2. We've done a lot of work with it, but we don't have any. We have some demo implementations that we can run, but we have not yet implemented it overseas. That's for going from WHONET to DHIS2. In the other direction, if you export from DHIS2 to WHONET, We haven't. It would be. We'd have to think about it. No one's really asked for it. No, actually, no. We did do it. We did do it for one of the projects. So that is, yeah. If somebody has something like that, we should be able to handle that. The problem with DHIS2 is that every DHIS2 implement, it's like Excel. You have Excel, I have Excel, but we didn't do it the same way, and it's even more complex with DHIS2. So it's very highly customized by the local and national users. DHIS2 has event modules, tracker modules, and data set aggregate statistics. It also depends on the kind of data. WHONET does not manage aggregate statistics. WHONET prepares aggregate statistics, but WHONET needs isolate level data. As long as I'm talking about it, I'm going to whonet.org. Webinars. So I'm going to shrink that. And here you can see, I mentioned earlier today, November 2023, we did a webinar on multi-drug resistance definitions. About antibiograms, I did talk about some of these features, and I did mention we do not have the ability to combine across classes. We can combine DISC-MIC test, which is very important, but we cannot yet combine between similar antibiotics. Some of the features I described today are here under WHONET installation configuration data entry. The focus of today was to try to focus on some of the non-human elements, but a lot of what I said today has already been covered in some of these other webinars. Our next webinar, my thought would be June, but because we're coordinating with FAO, we'll see what a convenient date would be for them. And the idea would be InFARM, the FAO correlate to WHO GLASS. And all the resources here, how do you enroll? What is the protocol? What are their accomplishments so far? Little plug for our next webinar. This will be a WHONET InFARM webinar led by us here in Boston with their collaboration. FAO also has their own schedule. They have a virtual learning center. I've participated and supported many of their FAO-led training initiatives. So if you work closely with FAO, you might hear about 2 trainings. This one, which isn't a training, it's just a 2-hour webinar, and then more specific, explicit, targeted training for the FAO priority audience. Okay, well, I do want to thank everyone. I don't know the mix of human, animal, food, environmental on this call. We started with human. We very much appreciate the input we get, but the value of the work on the human, on the non-human sector, because we are talking about food safety and risk factors and risk mitigation. And there are a lot of unknowns there. Is this practice that we do a good or a bad idea? A lot of times the evidence isn't there. How much does it cost in terms of animal health, food costs, production costs? And a lot of the existing data from high resource countries, very valuable. But we cannot simply say, well, it was the right decision for Denmark. We cannot say it's the right decision for some other country. So thank you for your contributions to that knowledge base. One thing we have seen with the Fleming Fund is a lot more closer coordination between laboratories, because a lot of countries have struggled buying discs, QC strains, and there's no reason to separate the human and the non-human sectors. They're buying the same reagents, they're buying the same QC strains, they still have to learn the disc diffusion method. So through the One Health, the Fleming Fund One Health initiatives, there's been a lot more coordination on logistics and knowledge that I think has been very impactful.